I'm Kaihre Brightwater. I go to New Mexico State University and I'm going into my senior year. I'm in the Polar Growth Lab. My mentor is Jacob Bouchier, but also Dr. Pamela Brown. It's her lab. What I'm doing is making deletions in the genome. So I'm taking out genes that will, you know how like a gene will give you blue eyes? So I'm trying to make deletions in the genome for Agrobacterium tumefaciens. So I'm taking out a gene that will, say, produce blue eyes for that bacteria. But the blue eyes in the case of the bacteria is a sugar production. So I'm changing the sugar production in bacteria to further understand what that sugar does. It's a pathogen, so we're trying to characterize it because it's not that well understood compared to E. coli, which is a common model for bacteria, or bacillus. Those are more common models for understanding bacteria, where this Agrobacterium, it's just less studied.
Would you shoot Bobby Riel's video?
So bacteria grow and divide, and when they are growing, they're building what we call the cell wall, and that's how they grow. And this cell wall is really important for them to be able to survive under a ton of different stressful conditions that they grow in, right? And so because it's so important for the bacteria to survive, it's kind of the go-to target for antibiotic treatments. And so things like penicillins and ampicillins are a class of antibiotic that specifically targets that cell wall and inhibits it from being constructed. So that's how it kills bacteria. But bacteria have developed tons of different ways and strategies to be able to combat treatment with antibiotics that target that specific structure. And so I'm really interested in understanding how the bacterium that we study, Agrobacterium tumefaciens, responds to treatment with those antibiotics. And Agrobacterium tumefaciens causes millions of dollars in crop damages across the world every single year. And so identifying specific ways in which it responds to treatment with antibiotics is super important, right? Because if it's able to evade all the antibiotics that we use, then there's no way to control its damage to crops.
So I really like being able to focus on one thing at a time. I feel like it makes me a lot more productive. It really helps me being in this setting for sure. In my home institution, a struggle is just time in general. You know, you have your mind in 5 different places, and then you try and go do part of your project, and you know, your mentor might be out teaching a class. But here, you know, everyone's localized. You can get a lot done really fast. Jacob just has so much, he knows so much about the project and so much about the techniques that whenever I have like just the smallest question, he just makes it super simple. So that helps a lot, especially he's here so much, you know, over the summer. Having a mentor is very guiding. Definitely feel like I'm not in the dark when I'm like going through my project. You know, things oftentimes like I don't know what I'm doing. Like I'll overthink one step 4 times, but I can just go and get like a very simple explanation and be able to reproduce that explanation and have a greater depth of knowledge on that subject.
I'm primarily a grad student, so I do research towards my dissertation. However, because I'm a 5th year now, I take on the responsibilities of a senior graduate student. And what that entails is often a lot of training to newer graduate students, as well as kind of a primary mentor for a lot of the undergraduates in the lab. Not just Kaihre, but we have, I think last semester we had like 9 undergraduates as well in the lab. And so I take on a lot of that role, also participating in things like lab meetings and asking questions and keeping people on task. So I actually have the unique position where I served as a student in the summer program, I don't know, in 2017, I think. And so it was really a rewarding experience for me. But then when I came to grad school and had the opportunity to mentor other students in there, I remembered how great of an experience it was for me, and how it kind of set me on this path to pursue research, which I knew was particularly interesting to me. And so it's kind of like giving back for, you know, what a great opportunity it was for me. Seeing people like Kaihre growing up in the field and me being able to help out in whatever way I can, it's important to me to be able to give back. So it's really easy in research to get tunnel vision, right? we speak in a very specific way and have a very particular set of jargon that we utilize so I can just talk about something very, very, very technical, and Dr. Brown or other graduate students in the lab will have a clear understanding of what I'm talking about. But if I take that and I take that set of jargon and that same conversation, I move it to the general public or even to other scientists in other labs, they could have no idea what I'm talking about. And so one really unique perspective that undergraduates and other younger students bring to the lab is that they come in and they ask you, what's going on? What are you talking about? It helps us realize that, sometimes when we're talking, we're not thinking about being comprehensible. I don't know the word there. Making it easier for other people that aren't necessarily within our field to understand what we're saying. And that's really important for transparency in research, right? You want the general public to understand what we're talking about. You want other labs to be able to understand what we're doing. And so in some ways, that's really important. Also, just bringing a new perspective to old questions, right? I've been working on this same project now for almost 5 years. And having someone like Kaihre coming in and shaking things up with new questions that I've never thought of before, sending me new papers I've never read before, really has a new, fresh set of eyes on something that I may have been stuck on for a long time. So there's a lot of value in that as well. Yeah, and what's really unique about the summer research program in particular is that you really get an immersive experience. Oftentimes when students are doing the research throughout the school year, they're missing out on opportunities that you would get if you're much more immersed for full time doing research. So for example, if you're in a really tough chemistry course, that's going to take priority to you over doing something like research, because you don't necessarily need the research to graduate. You need that really tough chemistry course to graduate. And so we find that the students who stay in our lab over the summer tend to get a lot more done, be more focused, and really get immersed into what it's like to be in a lab.
It's the cutting edge research there is. It's the farthest research there is in this subject. So I think that's super interesting to be a part of. Succinylglycan is something that there's research papers on it already, but furthering the knowledge and understanding out there is just super cool to be a part of. And that's a super fun thing to try and do. It does seem a little draining. It's a lot of work to try and stay in the lead when there's other people also trying to do your research and then maybe put out a paper before you over the same thing. It's I guess we have the same research. But it's super cool to try and figure out things other people just haven't got to yet. So some things I wanted to and I have gotten are lab techniques for molecular biology. So I really like bacteria and being able to work with bacteria in this setting with deleting genes and running a thermocycler and doing different assays is super interesting, and things that I can apply later in research or later in life depending on what I want to do. And just gaining more confidence in research. I think that's super useful, especially when trying to apply to interviews and things of that nature. It just seems like a very beneficial experience.